thank everybody for coming. I'm going to briefly introduce today's guest. Chad Weldy will be speaking today. He graduated in 2007 from Huxley College of the Environment with his BS in environmental science. And so we're happy to welcome him back. Since then, he's been at the University of Washington in the School of Public Health in the program of toxicology, working on his PhD, which he'll be completing during the next year. And today he'll be speaking about his PhD work, which has been on the effects of air pollution on public health with a specific emphasis on diesel. So, thank you, Chad.
Great. Well, thanks for that introduction. And this really is a pretty exciting opportunity for me because when I was here, this building was not even built. I think last time I was on this specific area, I think I was playing Frisbee. So this is kind of cool for me. But yeah, so I'm going to talk about my work investigating really kind of the pulmonary and cardiovascular health effects of diesel exhaust and the inhalation of really of a broad class of, I guess, air pollutants of fine particulate matter. Because fine particulate matter is kind of represents there are many different sources. It can be composed of a lot of different compounds. So we're going to be able to talk about that and talk about some of our findings that we think are important with regards to public health and with regards to a lot of our current decisions with regards to policy and increases risk of exposure. So first I want to point out that there are many different sources of fine particulate matter. And oftentimes when we talk about, or when we say fine particulate matter, we say PM 2.5. And that refers to the diameter of the particle. So PM 2.5 refers to a particle of 2.5 microns and less. So basically all the particles that are smaller than 2.5 microns. And we can look at, say, this would be, I guess, more of a representation of within our region. Traffic and trucking related sources of diesel exhaust is the primary concern. A lot of our materials are shipped by trucks. They're operating on heavy diesel. And just with regards to the general public that use diesel engines, this is a primary source of diesel exhaust and particulate matter within the atmosphere. In the Northwest, our marine shipping is actually a pretty important component of this as well. These ships operate on either using kind of crude oil or sometimes diesel. And an important component to this is when they come into port, oftentimes they have the option of whether or not they're going to plug in and use the city's electricity or whether or not they're going to turn their own engines to generate their own electricity. And oftentimes it's cheaper for them to just burn their own fuel. And while they're sitting directly in port for, say, a period of 5 days, they could be burning their own fuel for that whole period of time. And for example, Seattle had recently, with following violations of different air quality standards, actually made it a requirement that if ships were going to come in, they have to plug in. But then that ended up diverting ships to other ports to save money. So this is actually a concern. Wood smoke within this area is actually a pretty big deal because we oftentimes, in these areas within the Northwest, oftentimes within mountainous towns, a lot of people are burning wood as their primary source of heat. And with burning wood comes wood smoke, which is largely fine particulate matter. And of course, I just put this up of the trains to suggest that this is this is a concern. This is a concern particularly in Bellingham within Whatcom County, but really all of everybody down the corridor of Washington right along the waterway where trains that with the proposed coal terminal would be shipping. So this is a concern, and these are all potential sources. So what's unique about PM 2.5, and one thing I try to kind of emphasize is that other than other air pollutants, which are gases, PM 2.5 are particles. They are particles in the air. And it's the size that really makes them unique. So the diameter is critical for where the particle ends up in the lung following inhalation. If you have a large particle, say greater than 10 diameters, or 10 microns, they're not considered to be what would be called the thoracic particles, the particles that would be inhaled into the lung region. They would just deposit into the nasal area, the oropharynx area, and then that would just be swallowed, and you're not going to actually target the lung. It's the smaller particles, lower than PM10, so 10 microns in diameter, that can actually reach the lung. But the PM10 are going to hit more of the airways, the larger airways, and not necessarily interact with the more important tissues of the distal alveolar sacs. So if you look at the smaller particles, the fine particles, Those are the ones that are unique in their ability to remain within the airstream and target the distal terminals of the alveolar areas to then where they can then reside. Whereas the other particles, once they embed within the mucus-covered membranes of the airway, through this mucociliary action of being pushed back up, you end up just swallowing it. So the smaller particles are unique in their toxicity in that they are they're not cleared, and they remain in the lungs where they interact with within resident cells such as macrophages, epithelium, these other cells that can mediate other inflammatory processes and change pulmonary function. So what are we really concerned about? You know, traditionally we've always thought, okay, you inhale air pollutants, you inhale air toxicants, the concern is to the lung. And that is true, that is a concern. There is evidence that definitely shows a lot of air pollutants can either increase asthma or exacerbate asthma symptoms, general airway inflammation, increased risk of lung cancer. These are important components, but when we look at the actual epidemiology that looks at the broad risk of mortality, we're much more concerned about air pollutants' effects on the cardiovascular system. So first I want to point out this kind of, I think, a very pivotal study that really launched a whole series of other studies within epidemiology after that. This was just now is referenced so often that it's referred to as the Harvard 6 Cities study. And in the Harvard 6 Cities study, what they did of 6 cities across the country, they looked at total particles over a period of about 2 decades, or maybe just 10, actually it was just 1 decade, 10 years. And they were able to correlate mortality rates to then the particle size, the annual averages. So these aren't looking at kind of spikes or anything like that. They're just kind of annual averages, and how are those annual averages associated with mortality? And obviously you can see there's, within these cities, like total particles, there are differences. If you look at then into the fine particles, you can say maybe the lowest would be 10, the highest is maybe like 30, 35, and there are differences in mortality. So then when you correlate that, they had what was a pretty striking finding in that the particles that were available to the public to inhale was associated with mortality. And if you look at total particles, so this is including basically all the particles, including the fine, but also including the larger particles, there's a correlation, but it's not necessarily that good. But then when you look at the fine particles, all of a sudden this correlation is much more in line, has much more of an association. So this is really kind of where the evidence is pointed towards, that the fine particulate is what we're concerned of when we talk about health effects and risk to mortality. Of course, there's been other studies since then. This was a much larger study looking at it was American Cancer Society cohort that looked at 500,000 mortalities across the US. And they were able to associate, again, annual averages and how those are associated with mortality. And what was unique about this is the cataloging data of the mortalities were much more specific. And so this made it much easier to be able to look at specific types of mortality with ambient particles within the air. So they were first able to look at just cardiopulmonary mortality. Of course, cardiopulmonary mortality is a very broad category, but they find a positive association. So you look from basically like 10 to annual averages of like 10 up to like 30, or maybe 25. And you see that there's with each 10 microgram per meter cubed increase in particles. So if you want to think of that thing as like 1 meter cubed, if you took all the particles and you weighed it, and it was at, say, 10 micrograms. So each 10 microgram increase was associated with a 6% increase in cardiopulmonary mortality. But what was great about that study is then they could then go on furthermore to identify specifically types of mortality and look at whether or not the associations were stronger within different types of mortality. And of those 500,000 mortalities, 45% were cardiovascular. So this, again, highlights the importance of cardiovascular disease within our society, that 45% of those are cardiovascular related. Only 8% were pulmonary. So upon further stratification, you can then look at those associations at a much more specific level. So all cardiovascular disease and diabetes, you see that for every 10%, or every 10 microgram per meter cubed increase, there's a 12% increase in risk of mortality. So if you think about that from that Harvard 6 Cities study where the lowest was at 15 and the highest was at like 35, so that's a 20 microgram per meter cubed difference. So if it's a 20 microgram per meter cubed, you double it. So that's a 24% increase in risk of cardiovascular mortality. But if you look at ischemic heart disease, so this is largely when we talk about heart attack, when we have impairment in blood flow within the coronary vessels that can limit how well the heart functions and induce myocardial ischemia and myocardial injury, then you see this increases up to 18%. So that same 20 microgram per meter cube difference would be a 36% increased risk of an ischemic myocardial event that would lead to mortality. And of course, it also is associated with dysrhythmias, heart failure, cardiac arrest. But what was important about this study was that it identified that when we talk about mortality, we're still focused on the cardiovascular system. When we look at effects, say, COPD and respiratory diseases, it's actually not associated or even in some ways protective. I don't think anybody's going to suggest that it's protective, but it may suggest that there's other things at play. But again, it focuses that the risk of mortality is the cardiovascular system. So this highlights, I guess, two different types of exposure. One is that when we look at annual averages and you're talking about decades of exposure, really you're talking about the cumulative exposure. Someone who, for every year, they're living in a place that has higher levels, and they kind of accumulate to a higher level of exposure. And then there's the difference idea that there are spikes in exposure, that throughout the year, particles in the air don't just remain constant. Obviously, particles, the concentration of particles may be incredibly high near a tailpipe. They may be lower away from the tailpipe. But an important aspect of spikes is that our weather can influence how the concentration of particles and the concentration of fine ambient particulate matter. So I want to point out the idea of mixing in our atmospheric mixing. It's called the adiabatic lapse rate, is what basically references to the air that's closer to the surface moving upwards. You know, you have typically the sun comes down, it warms up the air, or warms up the surface, which then creates a layer of warm air right near the ground. And the warm air, obviously hot air rises, so if you generate, say, diesel exhaust, it goes up into the atmosphere and it removes it. So you could have, say, a significant amount of particles being generated, but not for many people. Say the concentrations may not build up very much because there is a mechanism that our atmosphere removes it. That's why we have smokestacks. That's why we have those emissions are up higher, because we want it to get up into the atmosphere. But an important aspect of this is the idea of a temperature inversion. If all of a sudden it's colder near the surface and then a warm layer comes over, then that process of warm air rising is negated and you get this buildup pretty rapidly. So you can have this buildup of particles or whatever pollutants in the air that would lead to these spikes following temperature inversions. And of course, there are some dramatic examples of this. One would be the London fog incident. Now, this is kind of a famous incident, and one thing that strikes me about this is it was only 1952. It's not like this is, you know, going back hundreds of years of this like crazy event. This is just like 60 years ago, where it was a bitter cold winter, and in London, people were still burning coal. Coal is a major source of particles, not as much in the Northwest. If you go into the East Coast, China, coal makes an enormous source of their particulate within the atmosphere. But it was a bitter cold. They're burning a lot of coal, trying to keep warm, and then a temperature inversion occurred, and it basically trapped everything in there. And so if you look at the time, they were only measuring sulfur dioxide. But sulfur dioxide, what we know about this event is that this was largely particulate. But sulfur dioxide concentrations were measured and they can go up. And what they found is that you can see sulfur dioxide is creeping along, and then during that temperature inversion event, it spikes up. And then if you look at weekly mortality, coming around about 1,500, all of a sudden it spikes up to more than 4,500, 5,000 people in a week. So this is looked at of upwards of, basically, you would then say if this is our predicted weekly mortality, without this spike in air pollutants, you would expect that level to go up, stay there. So basically everything under that curve would be associated with the spike in air pollutants, the fog event. So this is estimated of upwards of 12,000 people died prematurely because of this event. And what's important to note is that following the spike, the level of mortality remained elevated for a period of time. And in some of these other extreme events, people have been able to look at mortality and recognize that mortality is elevated for upwards of 10 years. So there are different effects that can influence this. The biology of this is still not very known. We still are investigating potential effects that may influence our genetics that would then be carried on. But these are important aspects to it. So the question is, all right, we know that people were dying, but what were they dying of? And what was found from the London fog is that actually people weren't dying of extreme respiratory failure. People were actually largely dying of heart attacks. And so this has been further investigated with other more recent epidemiology studies. And looking at kind of temperature inversions and looking at spikes in air pollution that would be more reasonable to what we would expect in our local environment. And so this group, they're looking at PM 2.5 at real-time measurements, and then looking at how spikes would maybe be associated with myocardial infarction. That's our heart attack. And what they found is that with spikes in air pollution, for every 25 microgram per meter cubed spike is associated with about a 1.3-fold increased risk of an MI. So this is important because this happens within hours. So within 1 to 2 to 3 hours, this spike goes up. And then they found that these were elevated within about 2 to 3 days, and then they dropped down after that. There is debate right now. There is a recent publication, the British Medical Journal, that suggested that, that it doesn't overall increase our risk. These spikes don't overall increase risk, that the then risk of mortality drops afterwards. That's possibly just those who are at high risk of having an MI are just shifted forward. But I think that's still up to debate. So one thing I want to point out, though, is if we talk about, okay, those who are most at risk, we would want to say, okay, those who have pre-existing cardiovascular diseases would likely be most at risk. Pre-existing cardiovascular disease is about 1/3 of the population, though, or of the adult population. So 82.6 million American adults have one or more type of cardiovascular disease. If we look at those 76.4 million American adults have high blood pressure. 16.3 have coronary artery disease. So this is suggesting that a huge number of our population is already at risk, already has pre-established cardiovascular disease. And this could be an important component to this. If we look at actual acute cardiovascular events, although we would say it's only about 3 per 1,000 per year within men aged 35 to 44, it increases to 74 per 1,000 for ages 85 to 94. So this is suggesting that these risk of events increase with age. And there are many factors that can contribute to this, but it's likely that air pollution plays an active role in this. So first, I want to talk about, if we're not familiar, if the audience isn't familiar with kind of heart attack and what we mean by that, I can point out here that we're talking about a clot within the coronary artery that services blood to the ventricle. So I guess I'm going to go forward one just to point this out really quick for those who aren't familiar. So the heart obviously has atrium at the top, ventricles at the bottom. It pumps in this great unison that allows for deoxygenated blood to come into the heart, pump to the pulmonary vasculature, and then be able to return oxygenated and get out to the rest of the body. But the left ventricle has to pump really hard. You have a high blood pressure, especially if someone is hypertensive. And so first and foremost, the heart is a muscle, and it requires oxygen. And one thing that these figures tend to always leave out is the importance of the coronary vessels. That the coronary vessels supply oxygen to the heart directly in this very quick circuit. The right one's pumps right back. And so you get this highly oxygenated blood to come and service the whole heart. And there are certain vessels that would service different parts of the heart. So a heart attack, we talk about plaques in our vessels, and we talk plaques in our arteries. And that's a really important component to that. And I would argue that nearly every adult has some plaque buildup. It would be very rare to find a person who just zero plaque buildup. And having plaque buildup doesn't mean death. Having plaque buildup may mean you just get tired a little quicker. The blood can't quite get there as fast. But the real risk for an extreme acute event would be what happens when this plaque ruptures. Inside this plaque, which is composed of a whole bunch of different cells, a lot of cells that have taken up a lot of lipids, a lot of cholesterols, a lot of nasty stuff. And because there's not sufficient oxygen getting in there, it ends up, all these cells die. And you get what's called the necrotic core. And so the necrotic core within the plaque is this real nasty, soupy material. And if that ruptures out and leaks into the coronary vessel, that instantly stimulates a clot or an infarct. And so that's the acute event, when you have actually plaque rupture, which then limits blood flow. And then what you can end up having is ischemia, where there's not sufficient oxygen, which then leads to myocardial injury, and you end up getting dead heart muscle. So if you then look to this, if that left ventricle is pumping and it's now part of the muscle is dead, it's no longer to be able to pump as sufficiently. That's when you have ischemic heart failure. So an important component to this, an important component to the ability of blood to get through the coronary vessels and service the heart, is a concept called vascular reactivity. And it's the idea that our vessels dilate and contract to be able to, kind of in this dynamic feedback way, to maintain and set our arterial blood pressure. And our blood pressure is an important risk factor for having an acute event, as well as extreme events of enhanced contraction that can induce what's called a vasospasm, which would increase that risk of that plaque rupture and entering all this necrotic core. So this complex interaction of vascular activity, mediating dilation, mediating contraction, is extremely important. And if we look at, say, the vessel here, the inside of the vessel is called the endothelium. It's lined with these endothelial cells. And at first, the endothelium was thought to be not very important. It just kind of played this kind of passive role. You know, it was kind of the interface between the blood and the vessel. But ever since the understanding more about vascular activity and how the endothelium plays a role, we've recognized that the endothelium plays a critical function in mediating vessel dilation and mediating vessel constriction through the smooth muscle. So smooth muscle is another layer that goes around the endothelium. So if I want to point out a little bit more of the complexity of the interaction between the endothelium and the smooth muscle, is that the endothelium can mediate either relaxation or contraction locally at that certain site. But although it's very complex, and I'm not expecting people to come away from this knowing all the complex interactions, I would point out that a fundamental aspect of mediating vessel dilation is nitric oxide. Nitric oxide is a soluble gas. It's called a free radical in that there has a one extra electron on it. And it's synthesized within the endothelium. And once it gets synthesized, it's able to release out, and it's able to target smooth muscle, and that mediates vessel dilation. I'm going to point out here, you can just kind of get an idea that there's one enzyme that synthesizes it. So it's a protein that's able to, when activated, able to stimulate the production of nitric oxide, and it's called eNOS, or endothelial nitric oxide synthase. So we've talked about kind of the epidemiology and suggestions of the risk of mortality, but there are many limitations to epidemiology. First of all, epidemiology is suggesting a kind of a correlation, but is not actually suggesting a causative association. So what we need to know is, is the biology there to suggest that that epidemiology is true? And that's an important aspect to it. So several groups, including the University of Washington, have developed controlled PM2.5 exposure facilities where we can do exposures with animal models or human models, or with human volunteers, and we can investigate the biological effects. So this is, I work within the University of Washington Discover Center, investing in cardiovascular disease and traffic-related air pollution. And we are basically aimed at understanding the biology, understanding really what's called the biological mechanism of effect, and to predict, I guess, some of the important aspects would be understanding whether or not there's a biological plausibility, estimate actual concentrations that would be safe or unsafe, or at least add data to the risk assessment literature so we can understand more about it. And then also to predict potentially sensitive populations. We want to know who may be more sensitive. And one of the ways we can do this, by understanding the mechanism, we can predict certain genes that may play an important role in this. So just to kind of have a schematic of our facility, we have our diesel engine exhaust, our diesel engine, and then it's appropriately diluted with filtered air, so we can have a very controlled and very, in a dynamic feedback way, having a controlled system where we're exposing either humans or animals, and we can test biological function. So there have been many different findings within a pretty broad literature within diesel exhaust and testing in cardiovascular function. But I would say the most important finding that has been continually coming up is that diesel exhaust causes impairment in vascular reactivity and vascular function. So I want to point out one study that was done by another group, not at the University of Washington, that exposed healthy men to diesel exhaust at a concentration of 300 microgram per meter cubed for just 1 hour. They had them do some exercise. You know, they're going on the bike for a little, like 15-minute intervals, but just a 1-hour exposure. And 300 microgram per meter cubed, to put that in context, you know, we say like the annual averages between like 10 and 30 for some of those cities in the Harvard 6-city study. If you're standing behind a tailpipe, you can easily get over 1,000 microgram per meter cubed. And there's actually been human studies where they asked volunteers to walk next to a roadway and have monitors of testing what types of concentrations they may get, and they'll easily see next to 1,000, 1,500 microgram per meter cubed. So I'd point out that this is high, definitely high for like an annual average, but is actually for a short-term exposure is not that high. And what they were wanting to know is how well the blood vessels dilated. And they did that by infusing different drugs that stimulate vessel dilation. And they measured what's called the forearm blood flow. So they're basically looking at how well does the blood get through the arm following addition of these drugs. And what they found is that for the filtered air group, so just people who didn't get diesel, just filtered air, you know, you infuse, say, bradykinin and acetylcholine. These are molecules that activate eNOS, activate nitric oxide synthesis, and blood flow increases, goes up. Those that were exposed to diesel exhaust, all of a sudden it did not go up as much. So this is a significant difference, suggesting that just that 1-hour exposure impaired how well your vessels produce nitric oxide, which is an important risk factor for an acute myocardial event. We look at sodium nitroprusside, which is basically a molecule that's just dumping nitric oxide. It's just give the vessel nitric oxide, so it's a difference of stimulating nitric oxide production versus just giving nitric oxide. And you still see this impaired function. So this would suggest that it's not only impaired production of nitric oxide found here, but actually the smooth muscle seems to have some impaired function as well within these short exposures. And this was observed upwards of 24 hours after that 1-hour exposure. Work that was done at the University of Washington did a 2-hour inhalation with human subjects looking at 200 microgram per meter cubed, so less than what was previously shown, but 1 hour longer. And this was in the absence of any exercise, in the absence of any drug infusion or anything. They were just looking at what the vessel, the brachial artery coming down the arm, how the brachial artery diameter looks following inhalation of diesel exhaust. And they found that with a 2-hour exposure, the brachial artery diameter decreased. So basically your blood vessel constricted. That's an important risk factor for things like, the idea is that the brachial artery may not be that important for really setting blood pressure, but it's a good model for predicting what may happen throughout the body, what may happen throughout vessels, say on the coronary vessels on the heart, and that's going to be a really important thing that we really do want to know. So this is an interesting question. So how would inhalation of particles into the lung then cause your brachial artery to constrict? So that, obviously, that's not necessarily intuitive. We have to kind of figure that out. And this is a topic of intense debate. One thing I want to point out is that diesel particulate is a known pro-oxidant. And so to put that in context, I assume most people have heard of the idea of antioxidants, right? So we take antioxidants, we want to take antioxidants, or if we're not taking antioxidants through our supplements, we want high antioxidants within the foods we eat. So basically, antioxidants are to combat oxidants. And so we can think of it in that sense. So one idea is that, well, particles may actually, once you inhale them into the lung, may directly translocate into the vasculature. And there is actual evidence that small particles are able to, once you inhale into the lung, get into the vasculature, and it's possible that those particles are then targeting the vessels, and that's mediating the effect. Another thought is that PM2.5, when you inhale them, because of this pro-oxidants, can actually stimulate inflammation. And then you generate basically pulmonary inflammation that would then spill over. You basically get this generation of inflammatory cytokines. These are really important molecules that signal inflammation throughout the rest of the body. And that both of those could potentially target the vasculature, and both of those could cause what's called oxidative stress. It's basically, keep thinking of it as the idea of antioxidants, and this is trying to prevent, antioxidants try to prevent oxidative stress. And then that would stimulate whole body effects. So I guess I'm going to talk quickly about some of our initial work where we were asking that question of whether or not the particles versus the inflammatory factors would have a role. So there are two different methods that we wanted to first use to look at this. One was the idea, well, we can just do this in vitro and kind of test some different things. So in vitro meaning like literally in the glass, that we're actually taking cells in culture, and we can, after collecting particles, expose those cells to particles and see how those particles change biological function in the cells. The other idea is that we can test it ex vivo, meaning that we can actually take from a mouse, take the aorta, the aorta being the large vessel that's a conduit artery that services blood throughout the body, take the aorta from the mouse and section it in a way that we can test how well the aorta functions. So that could be an important model for testing how well cardiovascular, how well the vasculature may function throughout the body. So the first idea is, okay, well, if you inhale particles and the particles get into the lung and they translocate to the vasculature, what would happen if those particles are coming direct in contact with the endothelium, and the endothelium being the innermost layer of the vessel? And so we can then test the look at the different biological effect that happens. The other idea is like, well, you know, if you inhale particles, there are a lot of what are called macrophages in the lung, called these alveolar macrophages. And macrophages are scavenging around and they eat up particles. And if they eat up particles and they generate inflammatory factors, those inflammatory factors may then spill into the lung and change how well the endothelium functions. So we can test that by either taking endothelial cells in a culture dish, put diesel particles on them, and see, and then test for biological effect. The other is that we can have those endothelial cells in a plate, and then on top of those grow macrophage cells. And then with a membrane in between the two, we can treat macrophage cells to diesel particles and basically allow all the other soluble factors, all the other inflammatory factors from those macrophage cells can pass through the membrane and target the endothelium, but the diesel particles wouldn't. So we can basically isolate exposure from just the inflammatory factors of either the inflammatory factors stimulated from diesel particles or the direct diesel particles. And we can see and make judgments about whether or not one would be associated with impaired cardiovascular function. So before we had mentioned eNOS, so the enzyme responsible for synthesizing nitric oxide that's really important for dilation. And if we think that nitric oxide is impaired with diesel inhalation, then we may suggest, well, maybe eNOS actually just, there isn't as much of it. Maybe the transcript actually goes down and the number of eNOS enzymes actually decreases. But one thing we find is that if you treat endothelial cells with diesel particles, eNOS actually goes up. So this may suggest that actually this would be opposite. And when you're increasing eNOS levels, that may suggest there would be more nitric oxide available. This enzyme here, iNOS, or inducible nitric oxide synthase, is basically a marker of inflammation. And this would suggest that the particles do have an effect on endothelial inflammation. But this is limited to iNOS. Down here, this is MCP1, or monocyte chemoattractant protein 1. This is very important for the progression of atherosclerosis, very important for the progression of plaque buildup. And one thing we find is that the diesel particles basically don't do anything to the MCP1, maybe just a little bit. So this would not necessarily fit in our model of, well, would this be associated with impaired vascular activity? But when we expose the diesel particles to the macrophages, and the macrophage inflammatory factors then target the endothelium, we actually see an opposite effect on eNOS. The inflammatory factors actually downregulate the expression of eNOS. So that would be consistent with our idea that inflammatory factors may be mediating the impaired vascular function. iNOS also goes up, so this is suggesting increased inflammation. And also, importantly, MCP1 goes up pretty quickly too. So this would be an important factor that diesel particles, when they're exposed, when macrophage cells within our body would possibly be exposed to diesel particles, their inflammatory factors may mediate other effects in the endothelium that may be important for the progression of atherosclerosis and impaired vascular activity. But one thing I want to point out is that that's what's called gene expression, or looking at basically how well the gene is turned on. And that doesn't necessarily mean that the vessel wouldn't function as well. So we can test actually how well the vessel would, so we actually need to test what would be considered the vascular activity. We need to assess that. So one idea that we'll question, okay, how would we test that? We can take out the aorta from the mouse. And one idea would be, okay, if we want to test the idea, well, maybe the particles change how well the vessel functions, We can take the aorta and just treat them to diesel particles. The other idea is we can take the particles, expose them to those macrophages, and all the inflammatory factors get generated. And we can take that media, and from the media, we'll spin it down, filter it out, no particles in it, and we can expose that media to the aorta. And we can test how well the aorta functions from there. And we use this technique called wire myography. And it's a way to be able to vascular myography studies are able to measure how well the vessel functions with following treatment with different drugs. So this would be our myography setup that has wires so we can go through the aorta and we can treat with different drugs and measure really the force that's generated following either contraction or relaxation. And one thing we see is that if you take those rings, those aortic rings, you treat them with diesel exhaust, and we treat it with acetylcholine, which is stimulating eNOS, stimulating nitric oxide production, the diesel particles don't really have any effect. So as you would expect, acetylcholine stimulates relaxation, nitric oxide production, and the diesel particles don't do anything. But if you take those aortas and you treat them with the media that was stimulated with the diesel particles, then all of a sudden you see this impaired relaxation. So this is suggesting that the inflammatory factors, as we observed within kind of gene expression studies, stimulate vascular inflammation and seems to be a downregulation endothelial nitric oxide synthase, seems to also impair vascular activity, impair the ability to synthesize nitric oxide. So again, if we were going to look at this complex interaction that's there, and nitric oxide being the most important, especially with acetylcholine that stimulates through its receptor and calcium-mediated activation of the eNOS and nitric oxide, this is what we're really questioning. So what would be happening here? Is it actually just a decrease in eNOS enzyme level? That's possible, but it's actually not necessarily likely. If we look at the enzyme eNOS, it's a unique, or I guess not necessarily unique, but the structure of this enzyme is incredibly important. It's this homodimeric structure that is held together by these two cofactors called, labeled here as BH4, they're called tetrahydrobiopterin. And these are really sensitive to oxidation. So normally, you have your electron that's donated from NADPH, it's transferred from its reductase to its oxidase domains, and you have with L-arginine and oxygen, the generation of nitric oxide. So this is when it's operating normally. It's just activated, pumps out nitric oxide. But if, say, you get oxidants, in this case I'm showing superoxide, this is basically oxygen with an extra electron, you can get oxidation of these tetrahydrobiopterins. So instead of having sufficient copies of this dimeric structure, you can oxidize those, leading in monomers rather than dimers. And when you reduce these, or you oxidize these cofactors, and it's no longer in its proper structure, when an electron is transferred across, it is not able to participate in this reaction down here. So what happens is you actually the electron still is transferred, and it interacts with oxygen to generate more superoxide. And this is what we would call a feed-forward mechanism. That once you have the initial generation of oxidants, an initial oxidation of eNOS, it actually just is a pro-oxidant itself, further exacerbating the problem. So I think with some of our in vitro and some of our ex vivo studies, it's looking more towards the idea that the inflammatory factors may be an important part of this. So it's the idea that you inhale diesel particles, uptake within macrophages and endothelium, you have airway inflammation due to oxidative stress, those cytokines enter the systemic circulation, and then you get vascular oxidative stress impairing vascular function. This is going to bring me to some of our other work that we're investigating. Basically, how does our body defend against that? If we have oxidative stress in the lungs, oxidative stress in the vessels following diesel inhalation, how does our body protect ourselves from that? And we talked about antioxidants. And antioxidants in the diet are an important source of antioxidants. But I would argue, more importantly, our body produces its own antioxidants. One of which is the antioxidant glutathione. Glutathione is a good antioxidant in that it has the sulfhydryl region. It can participate in redox coupling. But it also participates in a lot of other reactions. It can be found in upwards of millimolar concentrations in certain cells. I mean, it is extremely high levels in cells. It's the principal molecule that sets how well our cell is in a reduced state. It is synthesized in a two-step process, one of which the rate-limiting step is carried out by the enzyme GCL, or glutamate cysteine ligase. And GCL is composed of modulatory and catalytic subunits. And so basically, so there's two genes that when they're activated and when they produce their protein can then lead to this production of GCL. And that activity, the expression of these genes, are important for regulating the activity of GCL and then the subsequent production of glutathione. So if we were to look at this again, one theory we have is that, well, you know, oxidative stress is likely playing an important component to this. Our antioxidant defense system is also likely playing an important component to this. So then glutathione and our body's ability, our cell's ability to synthesize glutathione may play an important role. And it may prevent that. The reason why we have highlighted on those genes, the GCLM and GCLC, and why we think this is important, is that GCLC and GCLM are both what are considered highly polymorphic. And that means that basically we can go back maybe thousands of generations or something like that, and somebody may have had a mutation in their DNA code. You know, we have our normal DNA code, and if one gets switched to another, a different DNA base, it may not be a problem. You know, in some cases, that one DNA base change may be a very important problem, and it may lead to embryonic lethality. But sometimes it may just not be that important. And so then different lines are going down, you get more, it just gets passed down from one generation to the next. And so that would also be considered what's called a single nucleotide polymorphism, basically a mutation that's common in more than 1% of the population. And sometimes those polymorphisms and those mutations are not necessarily overtly bad, but they are associated with increased risks of disease. Both of GCLM and GCLC, if you have one copy, so you have for each gene you have two copies. If one of those genes happens to have that one mutation, you have a twofold increased risk of having a heart attack. If both of your genes, both of your alleles of that gene have that SNP, then you have an eightfold increased risk of having a heart attack. And that would be important because about 1 in 5 of us has this SNP. So this is suggesting that obviously our genes are different from one to the next, and these SNPs may play important roles in our disease progression. Then when you look at these people that would have the SNP, either having one copy or two copies, they have impaired response to acetylcholine. So this is actually in measuring in coronary blood flow and infusing acetylcholine into the coronary arteries and looking at dilation in coronary blood flow, that it's impaired. Those with this SNP are associated with impaired blood flow. Again, this is kind of more epidemiology, and this isn't mechanistic. Like, we don't know for sure if that's what's happening. But this would suggest that having this SNP and not being, what's known about having the SNP is that when you get oxidative stress and you need to upregulate the expression of those genes, GCLM and GCLC, that you can't upregulate, they don't upregulate as quickly. And so you end up having impaired glutathione synthesis. So this may suggest that impaired glutathione synthesis is very important for cardiovascular disease. So we're interested in this, and the way we've gone about investigating this is looking at in a mouse model of compromised glutathione synthesis by targeted disruption of the GCLM gene. And so we have generated what's called the GCLM knockout mouse. And by lacking the modifier subunit, the GCLM, the GCLC that's remaining there is just not very capable of producing glutathione. So to put this in context and the importance of glutathione, if you knock down GCLC and you have no glutathione, it's embryonic lethal. You have to have some glutathione there for normal biological function. The total knockout mouse, so again, you have two genes, and if both of them are disrupted, then you have about 10 to 20% of normal glutathione levels. So that's severely reduced. And you have what would be called the heterozygous mouse, and that you have normal two genes. That'd be one is knocked down and the other one's still functioning. You have about slightly lower, maybe about 80 to 90% of normal glutathione levels. And so we think that actually the heterozygous mouse may actually be an interesting model in that it's somewhat similar in some ways to the human polymorphic condition, in that a person with that polymorphism doesn't have zero glutathione, isn't walking around with both copies of their GCLM knocked down. They just have an impaired ability to activate it. And that's kind of what the GCLM het is, the heterozygous mouse, in that they have one functioning copy, but the other one isn't as able to work. And what we know is that you need both copies of this in the mouse model to really have sufficient levels. So what we first wanted to know was if we think inflammation is an important component to this, would mice with compromised glutathione have an increased risk of pulmonary inflammation following exposure to diesel particles? And so what we had recently shown is that if you are heterozygous, and that the mouse, again, has one functioning copy, one not, they're more sensitive to diesel particle-induced lung inflammation. So this is suggesting that, so we did this through collecting diesel particles and instilling them through the nose to be able to directly target the lung. And then we assess lung inflammation by lavaging it, rinsing out the lungs and collecting cells. And we can then look at how the level of neutrophils, so another important acute inflammatory cell, how well neutrophil influx into the lung, and also looking at inflammatory cytokines. So those same molecules, those same proteins that we think are important for mediating vascular function. So we showed that if you're heterozygous, the mouse are heterozygous, they're more sensitive. And if this happens to be somewhat similar to the human polymorphism, it may somewhat suggest that those with that polymorphism may be more susceptible to air pollutants or to diesel exhaust. So obviously we recognize that there's many limitations in this. This is a mouse model. This is not humans. But I think what it does is suggest that these genes may be important in this. But of course, what we were doing there was testing lung inflammation. But really, we want to know actual vascular activity. The vascular activity is going to be important. So what we did was a short-term exposure, in that we exposed them to diesel exhaust for 6 hours at 300 microgram per meter cubed. And then the next day, we sacrificed the mice, and we looked at vascular activity. What we found when we look at acetylcholine-mediated relaxation, this is just in comparison of wild type and the heterozygous mouse, because we think that the knockout mouse, as interesting as it is, and that it has extremely low glutathione levels, is not necessarily as representative as a human condition. So we want to just focus on what we think is more representative. And we showed that the diesel exhaust didn't seem to change the acetylcholine-mediated relaxation. If we compare within the genotypes, we see that maybe the heterozygous mouse was slightly impaired, but that's not very significant. That's not going to be very much. But what's striking is that we found that the aortas were highly sensitive to phenylephrine-induced constriction. So this is epinephrine or adrenaline. This is our body uses this to be able to mediate vessel constriction through our sympathetic innervation of our vessels. And what we found is just that 6-hour exposure increased sensitivity to phenylephrine-induced contraction. If we looked at the heterozygous mouse, we see that same thing, but it seems to appear that the heterozygous mouse, if we look down here, the het versus the wild type, that the heterozygous mouse are much more sensitive to the diesel-induced changes in vascular sensitivity. I guess I want to point out that this is important in that we use, as I mentioned, our vessels are innervated, and that means that we have neurons that are going to our vessels. And I always like to kind of point out, like, when you stand up in your chair, if you're used to, you get the head rush, the only reason why you don't just like keel over and fall down right then is that your body is stimulating vessel constriction in your legs to keep blood pressure up in your head. And so I guess what I point out is that our body stimulates vessel constriction all the time. And if you're more sensitive to vessel constriction following diesel inhalation, that's when you may be more at risk of, say, that vasospasm or these increases in blood pressure that may be important risk factors for an acute myocardial event. So I guess what I want to say in conclusion, I think we are suggesting that inflammatory factors from diesel-treated macrophage cells cause endothelial inflammation and impair vascular activity. Of course, that was done in vitro and ex vivo, and that's not necessarily a human, that's not necessarily the mouse, so this is just kind of ideas that we can take from that. We show that mice lacking one copy of GCLM show increased sensitivity to diesel-induced lung inflammation. So this is suggesting that if that was going to be suggestive to the human condition, may suggest that humans would be more sensitive to lung inflammation as well. And that aurors taken from mice exposed to diesel exhaust have increased sensitivity to phenylephrine-induced contraction, and that it was exacerbated in the heterozygous mouse. So further suggesting that these genes we think may be important in mediating our response to diesel inhalation. Again, there are many limitations to this, but I think this is important because even if those polymorphisms are associated with slight changes in sensitivity, it's 20% of the public. So you could have 20% of the public that would be more sensitive. And I think when we consider our regulatory policies, that's going to be important. So I guess we want to kind of this, really, when we talk about short-term spikes and short-term exposures, we're talking about our risk of an acute myocardial infarction. But there are many things that can influence this, one of which is vascular activity. So how will our vessels dilate and contract? And that can, impairing that can increase our risk of an acute MI. What's called our vascular oxidative stress, we think is an important risk factor for changing vascular activity, which would then suggest an increased risk of acute myocardial infarction. And there are many factors that can change vascular oxidative stress. Particles in the air may be one. Other environmental factors could be one. And our genes, I put up here, GCLM SNPs, that may be one aspect of the genetic factors that would influence vascular oxidative stress, all of which increasing one's risk of acute myocardial infarction. So I guess kind of the translation and communication component to this is that we do believe that our genes influence our response to environmental factors. I'm not saying that we have definitive evidence that this is what's happening, but this suggests that our genes do influence our response to environmental factors. GCLM SNPs are frequent and that 20% of the public has them, and they may influence our biological response to particles. And regulatory policies need to take this into account. If we're setting regulations to influence the whole public, but we aren't recognizing that certain subsets of the population are more sensitive, we need to be aware of that. So I just want to give thanks to Dr. Ruth Sofield, who was kind enough to invite me up and give this talk. And of course, Western and Huxley College, which gave me my undergraduate training and was able to launch me into a great graduate program at University of Washington. My advisor is Dr. Terry Kavanaugh, and all the other Kavanaugh lab members helped me out with this. We collaborate with down in vascular biology at UW, who helped me out with wire myography. And our civil engineers that we work with, Dr. Tim Larson and Jim Stewart, who help out with the diesel generation. And I'm with the NIEHS Discover Center and the Environmental Pathology and Toxicology Training Grant that I'm a member of. So I guess with that, I would take any questions. Yeah, actually that is a concern, because when wood is combusted, it is not a complete combustion. And that when a complete combustion would suggest when you burn something like a carbon-based source, you would reduce everything down to carbon dioxide and water. And when it's not complete, and as you have it in your fireplace and you have it, then you result in other things that would be carbonaceous in nature, and there would be particles. And oftentimes when you're combustion of wood smoke, you get particles, and a lot of those can have pretty toxic compounds on them. And wood smoke is considered a carcinogen, or is thought to be a carcinogen. And a lot of that has been suggested to be an important aspect of this. And actually, there's a considerable effort in certain towns that are much more sensitive to this. Because certain areas will have everybody burning wood smoke. And then if you have an inversion coming in, then you can have really high levels really quickly. Yeah.
So actually,
are we running out of time?
We have a class coming in here. So what I would suggest is if you had a question for Chad, if you'd be willing to take it outside.
Yeah.